Hello class, how are you all? I hope you all are doing good. In today's online lecture series, I'll be taking the topic of hemoglobin. This is a topic from MSc Chemistry, Organic, Semester 4, and I am Dr. Shachi Mishra, Assistant Professor, PG Department of Chemistry, Jayaprakash University, Chhapra. So let's start. But before that, please note my email ID shachicdri2013@gmail.com, and for any query, I repeat, for any query you can WhatsApp me on this number 8707448054. So in today's lecture, we'll learn about the structure of hemoglobin, function of hemoglobin, and synthesis of hemoglobin. First of all, what is hemoglobin? It is a protein found in red blood cells, and it gives red color to your blood. It is involved in transportation of O2 from lungs to tissue via blood. Now you must be wondering why O2 is required. See, O2 in tissues is required to drive oxidation of metabolic fuels, which are food that we eat to get energy. Hemoglobin is synthesized inside immature erythrocytes, and it consists of two components, heme plus globin. In an adult, hemoglobin A is the most common form of hemoglobin. Now first take heme. Heme is a prosthetic group made of hydrophobic porphyrin ring. Prosthetic group is a non-protein part that is combined to the protein. So heme is prosthetic group made of hydrophobic porphyrin ring. Fe2 plus is complexed in the porphyrin ring by two main chain histidine residues. The organic portion of this heme molecule is protoporphyrin 9, which is a circular system of covalently connected pyrrole groups which contribute these nitrogens in order to coordinate with iron. So this protoporphyrin 9 have 4 pyrrole groups, and this circular framework is appended with 2 vinyl groups, CH-CH2, CH-CH2, 4 methyl groups, CH3 here, CH3 here, and CH3 here and here, and 2 propionate groups, CH2, CH2, CO minus, and in this pyrrole group, CH2, CH2, CO minus. So when this protoporphyrin 9 is coordinated with this Fe2 plus, then heme is formed. So the only difference between protoporphyrin 9 and heme is this coordination of Fe2 plus ion. Heme complex is inserted into the protein with the propionate group protruding into the solvent. And the functioning portion of the heme is the tetracoordinate divalent Fe2 plus ion, which reversibly binds to oxygen. Now, globin. Globin is the protein part of hemoglobin. It is made up of 4 polypeptide chains in the quaternary structure. These are the 4 polypeptide chains. It contains 2 alpha chains, which comprises of 141 amino acids, and 2 beta chains, which comprise of 146 amino acids. Each polypeptide has a heme, and each heme group can bind oxygen. So in total, this globin molecule can bind 4 oxygen molecules. And this oxygen binding occurs in a sigmoidal fashion. This I will explain to you in detail later. But before that, you understand that each oxygen binding is in a cooperative fashion. What does cooperative mean? Cooperative means that once one oxygen atom binds to the heme group, binding of other oxygen groups to other heme groups is facilitated. This is known as cooperative fashion binding, and it is also known as allosteric effect. Now let us see what are the functions of hemoglobin. See, biological processes are complex and you need to have a very delicate balance, and so is the case with oxygen transport. So, why hemoglobin was required and what kind of binding was needed? Like we needed binding between iron ions and oxygen that must be strong enough that oxygen can be transported, yet it should be weak enough to provide a means for releasing of oxygen where it is needed. You see, the free ions in water, they form very unstable complexes with molecular oxygen, while the complex of oxygen with iron 2 contained in heme group is only slightly more stable. So what facilitates this binding in hemoglobin? These heme groups are tucked into the hydrophobic folds of the globin peptide chains. This hydrophobic environment, this whole hydrophobic environment, it fosters the formation of relatively stable complexes between the molecular oxygen and heme iron. So we can say that the hydrophobic environment of the heme group makes hemoglobin useful oxygen carrier. Now we'll check the oxygen binding curve. As I told you before that we needed such protein which should be strong for binding oxygen so that it can transport, and at the same time it should not have that much strong bonding that when we require oxygen it will not be able to release. So there should be a balance that when the pressure of oxygen will be low, it will release oxygen, and when it will be high, it will bind oxygen and thus transport oxygen. So for achieving this hemoglobin binds in sigmoidal fashion. This graph is sigmoidal in nature. So you know the fraction of oxygen in the peripheral tissues, concentration of oxygen in the peripheral tissues is a fraction that is there in the lungs. Means the concentration of oxygen in lungs is very much high compared to that of tissues. Here in this case we will be measuring the concentration in terms of partial pressure. So we can say that the partial pressure of oxygen in lungs is high and the partial pressure of oxygen in tissues is very low. So in the lungs, what hemoglobin does? It takes up the oxygen, means it gets loaded with oxygen, and then it transports it to the tissues via blood where the partial pressure of oxygen is very low. So you see what happens, that there is an equilibrium existing. Hemoglobin and oxygen, it combines to form HbO2, which we can say is oxyhemoglobin. So there is an equilibrium existing between Hb plus O2 giving oxyhemoglobin. So when the pressure is high, it facilitates the formation of HbO2, and when this oxygen laden hemoglobin HbO2 goes to the tissues where the partial pressure of oxygen is low, the equilibrium shifts towards the release. So from this graph you can see when the pressure is low, the loading capacity of hemoglobin is also low. That is, it is unloading the oxygen and giving it to the tissue. But here, suppose this point we are taking. At this point, the concentration, the partial pressure of oxygen is around 90%. So it is quite high concentration. So at this it will get loaded with oxygen. So you see it switches from 10% to 90% bound over. Approximately fourfold change in the oxygen availability. Suppose 20 was the oxygen availability over here and here 60. Means with fourfold change in oxygen availability, the loading capacity changes from 10 to 90%. Therefore, the protein that we required was to have effective unloading at low concentration and effective loading at high concentration, and we accomplished it by using hemoglobin as a transport protein. So let's see. Hemoglobin, it's a transport protein. Heme binds O2 reversibly. It has two states, oxyhemoglobin. It is the state in which the 4 oxygen molecules are bound to the hemoglobin unit, and the deoxyhemoglobin unit. Deoxyhemoglobin state in which no oxygen molecules are bound. Okay, so the red color of oxyhemoglobin you are aware, and in the veins you see slightly purplish color is there because it is carrying the deoxygenated hemoglobin. Okay, so hemoglobin can be either fully loaded or fully unloaded. It exhibits cooperative binding or allosteric effect. I hope this point is clear. I'll write it over here. Allosteric effect or cooperative binding is that the uptake of one ligand influences the affinities of the remaining unfilled binding sites. Now, let me tell you about carbon monoxide and cyanide poison. You see, carbon monoxide reacts with heme iron of hemoglobin to form a complex called carboxyhemoglobin. Okay, oxygen was forming oxyhemoglobin and this carbon monoxide forms carboxyhemoglobin. Now, this complex is about 200 times more stable than oxyhemoglobin. Breathing even low levels of carbon monoxide raises the amount of carboxyhemoglobin and decreases the amount of oxyhemoglobin in the blood, which results in carbon monoxide poison. Okay. Now cyanide ions, they can bind more tightly than either oxygen or carbon monoxide. And what they form? By binding to hemoglobin, cyanohemoglobin. So all the compounds that produce cyanide ions are deadly. You must have heard that we are advised not to eat seeds of several fruits. It is because they are containing very small amount of cyanide ions, and if we will have regular intake of those seeds, the cyanide concentration will increase and it will further lead to cyanide poison. Okay. Now let us see how hemoglobin is synthesized. You see, the synthesis of heme protein of hemoglobin, it takes place in mitochondria. It begins with the condensation of glycine. Glycine is an amino acid, and succinyl coenzyme A to form delta amino levulinic acid (ALA). Okay, glycine and succinyl coenzyme A, they condense to form delta amino levulinic acid (ALA). ALA then leaves the mitochondria and it forms prophylogenogen, and through a series of following reactions it forms coproporphyrinogen. Okay, then this coproporphyrinogen, it returns to the mitochondria and produces protoporphyrin. Protoporphyrin then combines with iron to form heme. Heme then exits the mitochondria and combines with the globin molecule, which is synthesized in the ribosome to form hemoglobin. This is an overview of heme synthesis. Like I told you, succinyl coenzyme A condenses with glycine. In the presence of ALA synthase, it acts as a catalyst to form delta amino levulinic It then exits the mitochondria and forms prophylinogen, and through series of reactions it forms coproporphyrinogen, which then enters the mitochondria and forms protoporphyrin. Protoporphyrin then combines with Fe2 plus ion to give heat. So that's all for today. If you have any doubt, you can ask me on my WhatsApp number which I have given on the slide number 1.